Good afternoon, ladies and gentlemen. I'm pleased to be here and to present some data and information about the human microbiome. And who in the audience, who ever heard about this microbiome? Please raise your hands and show me how many are. Oh, only few. I think so. That's good that I have the chance to talk about this microbiome, because this is a very important companion to you. And your microbiome are a lifelong passion, compassion. And he will have much efforts and much important effects on your life. In the medical literature, and the world-renowned medical literature like Gastroenterology, the most famous publication in gastrology, and Nature, one of the biophysical world-renowned literature, they have issued data on the human microbiome. And the last one, the current issue of Nature, titled on the cover with "Complex Diseases Get Complicated." And really, medicine is complicated too, but the microbiome is most complicated. Even in the media, this issue was published and was discussed, and the New York Times magazine in 2015 asked on the cover, "Can the bacteria in your gut explain your mood?" And look at these little guys beyond. These guys might manipulate your mind and your mood, and that's really astonishing. Some people say the human microbiome is the human superorganism, not the Superman, but the superorganism. And this delights what role plays the human microbiome in our life. So my agenda for this presentation is the human microbiome. What is it? Where is it? What is its function? And what is its correlation with disease? How can it be manipulated or perhaps cured? What's the therapy of these microbiome, and what will be the future research and the scientific development in the future? What is the human microbiome? A microbe, a tiny living organism such as a bacterium, fungus, protozoan, or a virus. And the microbiome is all of them, the collectively microbes in the human body, a community of microbes. And the biofilm, a community of microbes that live together on our surfaces, like the skin, like the mucosa, and anything else. And the human microbiome is what we we are human, but actually we are more bacteria than human, because the human metagenome, all genes of the microbiome, is more than 100-fold of the human genome, which was detected in 2003. And the microbiome was, in its molecular consequences, was detected in 2010. It is a friend and a foe, protective or aggressive, and it is very, very metabolic active. I will show it later. The components of the microbiome are trillions of biologic microorganisms, the microbes. In some, this is the microbiota. Bacteria, viruses, and fungi. These are the components of the microbiome. And some of the important characteristics of the microbiome are the diversity, the richness, and the abundance, forming the core microbiome, which is typical for one person. And here is some definition of these terms. The richness is the number of distinct members in the community of the microbes. The diversity, a measure of the richness and evenness characteristics of a community, often calculated a specific diversity index, which is very important for the healthy state of the microbiome and the healthy state of the person. And the dysbiosis, this is when the normal constitution of the microbiome is disturbed, then we call it a dysbiosis, the term used for a microbiota community associated with diseases. That's important for the explanation of disease states of the man. Here you see a picture which shows the core human microbiome in correlation with some influences, like the host pathobiology, the host physiology, the genotype of the host of the man, the environment, and the immune system. All these factors have effects in the human microbiome, and these are in correlation with the variable human microbiome, which can be affected by moving, by eating, by the nutrition, and by genetic disturbance or genetics situation of the host. This is a picture which shows all the bacteria. These are the microbiota of the gut and its phylae. The phylae are the main classes of the bacteria, and these classes, all these classes together forms the richness and the diversity. And if these are disturbed in the relation, so this comes to a dysbiosis, and this may cause diseases. So where is it, the microbiome? The microbiome is everywhere, in and out of a person. It colonizes the skin, the oropharyngeal cavity, the urogenital organs, and the main domain, the most part of the microbiome, is in the gut. Therefore, I will concentrate further on the gut human microbiome. Here you see the distribution of the microbiome in the GI tract, beginning from the mouth to the anus. And there's even a different distribution about the constitution and composition of the microbiota and the number of the microbiota. Most of the microbiotas resides in the gut, in the colon. Therefore, is the colon one of the important determinants of the function of the microbiome. And here you see an example of the regional differences around the world. These are microbiotas and microbiomes of children taken from Bukina, Faso, and from Italy. And by the distribution of the colors in these cake diagrams, you see that these are very different. So the distribution and the composition of the microbiome depends where you're living, in what environment you are living, and what genetic situation you have. And what's about the development? It's interesting and very important that the building of your microbiome starts at birth. And it depends on the way you have to come to Earth, whether you have a vaginal birth delivery or you have a sexual delivery. Childs who are born by vagina have the microbiome, the microbiota of the mother vagina, and child's born by sexual has part of the microbiome of the skin of the mother. So this forms the first time you get, as a human, your microbiome. And then it will be developed and stabilized in childhood. And this is depending, the development of your microbiome is depending on your personal situation, in social situation, family environment, and so on. And then in mid-adulthood, you get, so we think you get your core microbiome stabilized, and you develop the core microbiome which influences your own healthy state. And in the age, it becomes less, diversity and richness decreases, and therefore the microbiome can't protect against many diseases. And even if you get disabled and you need to have a wheeled chart. So you need to have this because your microbiome has been disturbed and has lose his protective properties. And that's a very important thing because some of our old-aged diseases are caused by an aging microbiome. So here you see a picture where shows the situation in the colon. You have a section through the colon wall, and up you see the inside situation. The first layer is the microbiome with this microbiota, and from this microbiota they're having the influences which are signaling remote function of the mucous layer cells, like killer cells, effector cells for immune reaction, for inflammation reaction, and even, so it's the actual research, for carcinoma development. So some data show that the development of the colorectal carcinoma, which is one of the diseases which can be really prevented by screening. So these development of the colorectal carcinomas can start with a diseased or dysbiosis of the microbiome. And here you see a picture showing the influences of the microbiome, which produces an intense metabolic situation. It's very productive in producing metabolites, and these metabolites passes through the mucosa and can affect the inflammatory response even in a pro-inflammatory or anti-inflammatory. So the microbiome can cause acting bad situation or can protecting against this situation. And this is shown how the gut metabolites in a circle influences the activation and the function of the health status of the host. And it is influenced by dietary and pharmaceutical influences. If you're taking drugs, antibiotics or anything else, the microbiome changes these drugs in metabolites which can be active or contractive against the disease or the health status of the host. And here you see one of the very important questions we ever had. What is the influence of diet to our health status? And the diet works over the gut microbiota with the brain-gut axis to the brain, about the liver axis to the metabolism of the liver, and by these ways they function as alterations of intestinal function like motility, sensory function, and barrier function. And what's very important, I will show you later too, that by these function, by these connection with the gut microbiota, mood disturbances can happen, like anxiety, depression, or autism. And here you see what effect we think have a stress. The primary question is, what is stress? Do you ever had stress? You might have influences of your microbiome, and this may develop in dysbiosis, and this dysbiosis is one of the causes of the functional gastrointestinal diseases, which is one of the worldwide most disease in gastroenterology. So the biologic effects, to conclude, are development of an innate and adaptive immunity, intestinal epithelial integrity, energy source, and the barrier to colonization against pathogens, microbial pathogens. And this is a list, not the last, it's a list of all the diseases and disorders which can be caused by a disturbed microbiome. And you see that's a very, very long list. And it's not only gastrointestinal diseases, there are even non-gastrointestinal diseases like diabetes, obesity, and metabolic syndrome, very important for the health disease of the society. So what will be the therapy and the therapeutic possibilities we have? We have the probiotics, we have the prebiotics and antibiotics, and the new one, we have the fecal microbiota transplantation. And here you see, that's the one of the last pictures, you see the preparation for a fecal microbiota transplantation. In the first you see a mixer. This is filled with stool. Then it's squirreled with saline. Then it's filled up in syringe. And this is put into via colonoscope in the patient. And that's the fecal microbiota transplantation. It's a very simple and a very simple method on very simple transplantation. So this is the take-home message for you. You have seen that the human microbiome and metagenome contain a vast majority of common bacterial species. An individual microbiome is quite stable over time. That's important because you hold this microbiome for your own personality, and this is only for your person. In comparison to healthy controls, alterations in the microbiota are recognized in a growing number of diseases, as I've shown you. And for the future, a better understanding of the functional interaction between the human host and the microbiome is very likely to lead a new diagnostic, prognostic, and hopefully therapeutic capabilities. Thank you for attention. Thank you.
Ladies and gentlemen, the next big question is a question of Shakespearean dimensions. To treat or not to treat with